Hello, my name's Culum and I'm a professor here at Macquarie University where I study fish cognition and behaviour. And today we're going to be talking about social learning in fishes. But before we start talking about social learning, it's probably best if we bust a few myths in relation to fish cognition. I think it's generally clear that most people in society today think that fish are incredibly stupid. You have this 3-second memory paradigm that you often hear, even on TV ads, which suggests that fish are so stupid that they can't remember anything for more than a couple of minutes. And here we have an example in a TV advertisement on a station in Australia, a TV station, which shows this goldfish swimming around a bowl perpetually, and every time it sees this brand of cereal, it remembers some new fact about that cereal. And I think the character from Disney's Finding Nemo really clearly illustrates that a fish could not possibly get by in the real world if it didn't have a half-decent capacity for learning and memory. And the reason Dory is so funny is that she can't remember her name, where she's going, where she came from, or anything like that. And clearly an animal working in a complex, real environment would have to remember those sorts of things, where food is, where predators are, where prey are, and those sorts of things. Well, of course, the scientific reality is that fish are much smarter than the public perception believes them to be. So what we're going to do is think a little bit initially about learning in fishes, and then we'll move on to social learning and think about culture a bit later. So this is one of the first experiments that I did as a young scientist in a lab, and you can quite clearly see here some evidence that fish are not only learning, but they remember for good periods of time as well. And this is an experiment where the fish had to find an escape route in a trawl net as it came down the length of a fish tank. And this is the number of exposures that those fish had to that particular trawl net. And you can see when we first ran the trawl over 5 separate occasions, they rapidly learnt to avoid that trawl net by finding the escape route in that net. We then set those fish aside for almost a year and tested them again, and you can see that their escape latency remains roughly the same as it was a year ago, and in fact they continue to improve. So it's almost as if they did 10 exposures to this test in a row rather than 5 separated by a year and then running another 5. So it's quite apparent that the fish are learning very quickly, and this particular species is a rainbow fish, and it only lives in the wild for perhaps for 2 or 3 years or something like that. Now everybody knows that fish live in schools or shoals, and you would think that, given that they are pretty social animals, that perhaps they have some sort of capacity for complex social cognition, complex social lives, and perhaps even social learning across a range of contexts. And one of the things that we know about fish shoals is that they're seldom made up of a random selection of individuals. And in fact, there's some work in guppies, for example, which quite clearly show that they learn to recognise individuals in their social group over a matter of 10 to 12 days. And some early experiments showed that in the lab conditions, fishes would gradually learn to recognise those individuals that they've been housed with over about 12 days. And if you compare to the amount of time they spent with unfamiliar conspecifics, that's in the black line here, you can see that they prefer familiar over non-familiar conspecifics, and that is maintained over several weeks. What you can also see over here is the preference after some 12 days or so of isolation, and in that case the fish had still maintained their preferences for unfamiliar fishes after 12 days. And in fact, another student conducted the same experiment some years later and found that in fact these preferences for unfamiliar individuals, familiar over unfamiliar, is maintained for at least 5 weeks of isolation. So once they've learnt the identity of particular individuals, they remember that information for quite some time. The other interesting thing is, as you would expect, as the group size gets bigger and bigger and bigger, their capacity to remember the identities of particular individuals declines. And you can see that by the time they get to a group size of about 60, they have difficulty differentiating familiar and unfamiliar individuals. But it does suggest that there are quite a large group sizes in which the benefits of familiarity might prevail. What are those benefits? Well, there are multiple benefits for hanging out with familiar individuals, both in a predation context, as we can see here, but also in a foraging context. Here we can see the mean shoaling index of, in this case, minnows in familiar and unfamiliar groups, and you can see that the shoaling index is higher, so they tend to hang out in more closely knit shoals. And this is the number of inspection visits of a predator, and you can see that groups of familiar fishes are much more inclined to inspect the predator to get some information about the identity and perhaps its plans for the future. And those are much greater in familiar groups compared to unfamiliar groups. So there's lots of reasons why you might choose to hang out in a group of familiar conspecifics. One of the other things we know about groups of fishes is that they tend to learn novel tasks faster when they're in larger groups as opposed to smaller groups. And this is that same trawl apparatus experiment that we visited just a few moments ago. And here we're comparing 2 fish versus 5 fish. And you can see the 5 fish learn very rapidly, similar sorts of experimental results that we saw previously. But the 2 fish really struggle. And in fact, it takes them a very long time to figure out that escape route. Part of the reason for this is that statistically there's a greater probability that a larger group of fish will find the solution to a problem, even if it's just by chance. But more importantly, from a social learning perspective, is that once one individual has solved this task, others can watch that and learn from that individual and hence solve the task themselves. And they also tend to follow one another, being schooling species. This is a similar sort of experiment. This one was done by Rachel Day and colleagues, and this is the same Rachel that gave you the 3rd lecture in this series. Here we trained fish to pass through a door and a partition in order to access a foraging patch behind that partition. In the first instance, the partition is transparent, and you can see here that the small group of fish take a lot longer to find that foraging patch compared to the large group of fishes. So this is the same kind of experimental result that we saw with the trawl. When you make that an opaque partition, however, it flips. So in fact, the large school do worse than the small school. And the reason for that is that that opaque screen is breaking the visual contact between individual members of the school, and they're much more reluctant to pass out of visual contact with their shoal mates and hence discover the foraging patch on the other side of that partition. So this strong social effect of exploration can have positive impacts with familiar large groups, and perhaps sometimes negative impacts in terms of finding new schools or locations or foraging patches, or perhaps even discovering the location of predators. So it can have positive and negative feedback implications. But one of the things researchers have been interested in for quite some time is in these social contexts where information is clearly passing through members of the group, are there leaders and are there followers? That's who follows who, and perhaps one of the things that might be influencing this is individual personalities. And that is exemplified here by this super green fish. Is obviously the odd one out and being the leader of in that social group. And it may well be not surprising that things like boldness, sociality and activity levels, these are all classic behavioural personality traits in fishes, All of these things in various arrangements have been found to influence social learning, and social interactions. In this particular experiment, we wanted to know if the personality of individual group members had a bigger impact on the dispersal of the group as a whole. And in this case, we picked 4 fishes at random, and we placed them inside the central compartment of this apparatus here. And what we're interested in is how long does it take them to figure out that there are neighbouring compartments that they can potentially discover. And of course there might be novel food patches and so on within those compartments. Now, prior to putting them in this apparatus, we ran a bunch of personality tests on all of these individuals, and we got 2 males and 2 females and put them in here. And then we just recorded how often they dispersed between these various tanks. And this is the result. And of course, one of the striking things is that each one of these different group combination of individuals had very different dispersal capabilities. You can see some of them dispersed a lot, and some of them, in fact, not at all. So what on earth is responsible for all of this variability? And one of the things that really stand out straight away is that this is not about the group average personality, nor was it some sort of consensus or conformity building within the group members. And indeed, what we found out was that it's the behavioural traits of individual group members that had a big impact on the behaviour of the shoal as a whole. That is, its propensity to disperse and discover these new locations. So firstly, the activity score of the least active member strongly explained the exploratory behaviour of the group as a whole. So this is the least active member effectively acting like an anchor. So they stay still, they don't move around very much, and that causes the rest of the school to disperse less. Of course, on the other hand, if that least active individual is actually quite active, then of course the school moves around a lot. The other variable was the sociability score of the most social group member, and this is about social cohesion. So when the social cohesion is very high, all the individuals are very social, and they tend to move together, and there's a great propensity to discover new locations and disperse into those neighbouring tanks. So taken together, it's pretty clear that particular individuals can have big influences on how information flows through particular groups. Now, some very interesting experiments was done by Webster and his colleagues, and in this instance they were working with sticklebacks, and what they showed was that fish can actually pool their knowledge in order to discover and solve novel tasks. And this is quite an interesting experiment. Basically what he did, he set up an experiment where the fish had to learn a 2-stage task. Some of the fishes were completely untrained in either of those stages. Some of them were trained just to one of those stages, and then there was a combination where some of the fish were trained to one and some of the fish were trained to the other stage. And so what happens is that those individuals that are trained to stage 1 and stage 2 can combine their knowledge and solve the task, the 2-stage task. In this case, the first task was to go towards, or be attracted towards, a light, and these fish had learnt an association between the light appearing and being fed. The others had been trained to enter into a feeding box, and the third group had a mixture of individuals. Some of them had been trained to approach the light, some of them had been trained to enter into the feeding box, and that combination clearly increased the probability that any naive individuals actually solve the task. So they're combining their knowledge to solve a new task. Quite interesting result from Mike. Now, clearly the behaviour of individuals within the shoal contributes to overall shoal behaviour, and fish are learning new tasks by watching and interacting with other individuals within the group. And this is the process which you now know is called social learning. And we know that information can flow about all sorts of different things that are happening in the environment through fish shoals and onto naive individuals. And this includes things like the identity and location of predators, what your mates might look like, the sexiest male or the sexiest girl or whatever, and perhaps even the identity and location of particular food sources. Now, this is one such example of social learning, and this is using juvenile Atlantic salmon. And in fact, these Atlantic salmon were reared in captivity in hatcheries. And in that hatchery environment, it's extremely artificial. So they've got no experience of predators or prey or what have you. And of course, when they're released into the wild, they have to rapidly learn these new behaviours or die. And one of the interesting things about these baby fish is that when you expose them to live prey for the first time, they actually have a fright response. They back away from them. So in order for them to survive in the wild, we thought we might try using social learning as a way to rapidly teach them to identify predators and prey. And in this case, it's the live prey that we're interested in. You can see here along the bottom line is a black line. Now these are individuals that have already been trained to identify and attack live prey items. And this is the kind of behaviour that we're trying to get hatchery fish to. This is the sort of behaviour you might see in wild fishes. As soon as a prey item arrives on the scene, they immediately grab it within just a few seconds. And that's the kind of natural behaviour we're trying to induce. The first pink line is this scenario here, where you can see a naive individual on one side of the compartment watching a trained demonstrator on the other side of that compartment, and that demonstrator is regularly attacking live prey. And you can see that these naive individuals that have that opportunity, that exposure to demonstrators, learn this task very, very rapidly in just 6 trials. And by 6 trials, that you cannot distinguish them from a pre-trained individual. This line here in green is what an individual on its own looks like if it's trying to learn this task by itself. And you can see it goes on, and it takes about twice as long for them to figure out that prey items are edible, sometimes 3 times as long. And so clearly social learning in this context is having a massive increase in the amount of time that it takes these fishes to learn the task. They're clearly learning the task much, much faster if they have that exposure to demonstrators. The other line here, which we haven't talked about yet, is this red line, and this is the negative control. So this line here is what happens when you have 2 individuals side by side and neither of them have any idea what's going on. And in fact, you get negative social feedback in that context. That it takes them a long, long time to figure out. So you can have positive social feedback and negative social feedback in these things. You don't want negative social feedback when you're training to prey items, but perhaps you do want negative social feedback when you're training them to predators, for example. This is another example of learning the location of a foraging patch. And here we taught the guppies to swim through a red door to access a foraging patch on the other side. And as we mentioned before, the familiarity of the group members can often have a big impact on the dynamics of the shoal as a whole. So here we have familiar and unfamiliar demonstrators. So some of the fish have familiar demonstrators illustrating how to take that foraging path, and unfamiliar demonstrators. And immediately you can see that those that have unfamiliar demonstrators take a lot longer than those that have familiar demonstrators. So that increased social cohesion is clearly helping. And you can see that illustrated here. This is the number of following events. And you can see that demonstrators are often following each other, and observers are often following each other. But this is particularly the case where those individuals are familiar with one another. So there's a lot more following going on in familiar groups. You can see what's happening over time in this particular graph. And the first thing you'll note is that when the individuals are familiar with each other, the observers are learning pretty quick, and the demonstrators are doing what they're supposed to be doing. They're clearly demonstrating this behaviour very, very nicely. And of course, the observers learn relatively quickly. But when you look at what happens in unfamiliar groups, there are two things that sort of strike you. The first is that the observers don't do very well. They don't improve much over time, and it takes them quite some time to learn this task. But have a look at the demonstrators as well. The demonstrators are also performing relatively poorly. And part of the reason that is because they're suffering from divided attention. They're not only having to solve the task, which is finding that foraging route through the red door, but they're also having to sort out all sorts of social interactions and hierarchies formation and those sorts of things. So they're distracted by the social complexity of the situation, and they're not focusing on the task at hand. So through a series of experiments over the years, we've shown that all sorts of interesting things can happen and influence the way in which information flows through social groups. And we find things like small fish tend to following other small fish, fish preferring following large groups rather than small groups of demonstrators, fish follow familiar rather than unfamiliar demonstrators, as we've just seen. Fish tend to follow demonstrators that have direct behaviour, that is, they're clearly motivated, and therefore it looks like they know what they're doing. And poorly trained demonstrators are often more effective than well-trained demonstrators, and that may seem a little bit strange, but sometimes the demonstrators perform the behaviour so quickly that the observers don't get the opportunity to see what's actually happened. Males will often follow females, but not the reverse, but that's a little species specific. And personality often defines the role that individuals are playing within each of these groups. And so what we can see is that the personality of group members, as well as various other social factors, often have a big influence on how information flows through groups of fishes. Now, fishes, of course, like other animals, including you and I, can make decisions based on our own private information or based on our observation of how others in the wider environment are behaving. And that's called public information. Now, Isabelle Coolin showed that the way in which individuals use social information versus private information can vary from species to species. And in her case, she was looking at 9-spine sticklebacks and 3-spine sticklebacks. And what she found there is that 9-spine sticklebacks tended to use social learning to find a novel foraging patch far more than the 3-spine sticklebacks. And one of the reasons we think that might be happening is that the poor old 9-spines are actually not very robust. They're kind of small, their spines aren't very big, they aren't very armoured, and they tend to be a bit more timid. So they hide away, watch what everybody else is doing. Once they've figured out what to do, they then go ahead and behave and make the moves for themselves. Whereas 3-spine sticklebacks are much more robust. They're built like tanks. They have big, strong plates on their body, they have much, much stronger spines, and they're just far less vulnerable to risk. So they tend to be more risk-taking, whereas the 9-spines tend to be more risk-averse. So these guys are quite happy to go out and explore by themselves because the risk is relatively small. Now, Jeremy Kendall showed that, in fact, sticklebacks are using what's called a hill climbing approach, which is sort of a local search algorithm to solve the task of how much private information or public information you should use at any given time. And basically what that means is that they try one approach and then they try something else that's just a little bit different. And if that improves their behaviour or the result or outcome or their fitness somewhat, then they'll try that thing again or some other local variant. And what that basically means is that it takes you to local hotspots of fitness. So in this case, you might find the hill over here. In this place, you might actually find that you go to some slightly suboptimal behaviour. But nevertheless, there's an opportunity to get across to the next hill using this technique. Now, we've seen from that prior example that the degree of risk-taking of different species can influence the proportion of public versus private information that they're using in order to make decisions. Now, what if we took that away from the species against another species sort of context and thought about it within an individual species context? So in that case, we might be thinking that perhaps individual personality traits might also determine the extent to which individuals use public or private information. So for example, a really bold fish might be quite happy to go out and explore a novel environment, discover things for itself, whereas a really shy individual might be less inclined to do that, more risk averse, and perhaps rely more on social learning. And that's exactly the question we asked in this experiment, except this time we were using guppies. And effectively what we did in this experiment is we trained a bunch of fish to go to a particular feeder. Now that feeder was always associated with a rich deposit of food. And some of the fish were trained to go to a purple feeder, some to green. Doesn't really matter which one. And we also tested the personality of every single fish that we tested in this setup. We then asked the fish, after several exposures, in fact 15 days of exposure, what is your preferred patch? And only those fish that actually got this right, that is they went to the rich patch, moved on to the next phase of the experiment. And in here we actually asked them a difficult question. We asked them basically, well, two really important things. The first is, what do you do if your public and private information coincide? So they're reinforcing each other, and of course in that instance you would expect them to go to the rich patch no matter what. And then what happens if the public and private information conflict? So you've been trained to go to one particular location where you expect to see a rich patch, but what happens if there's some public information that suggests that that information has changed, that in fact the environment has changed and the rich patch is now on the other side? Do you then use your private information or do you use public information? So you can quite easily set this paradigm up. You put your fish that's already been trained and you let them look at another group of fish that's feeding either on their expected location, that's the rich patch that they know from their own private experience is the rich patch, or perhaps they're feeding at the opposite patch that they've been trained to. And then we remove the social group and we ask the fish to make a choice. Do you go to the patch that you've just seen those fish at, or do you make a decision based on your own prior knowledge? And what we found is that the boldness score of particular individuals had very big influence on whether they used public or private information, just as we expected. Here you can see the situation where public and private information coincide. The bold fish are ignoring their private information and they're going to the poor patch, which means they're avoiding the rich patch basically. Shy fish, on the other hand, no problem at all. They go to the rich patch as demonstrated, but also according to their own private information. So the bold fish are exploring, ignoring social cues, and even ignoring their own experience, and they're trying something else. And the shy fish are just following their own experience and following the shoal. When the private and the public information are in conflict, the bold fish go to the rich patch, so they're avoiding that social information. So they're actually paying attention to their own private knowledge there. And similarly, the shy fish are doing as expected. Instead of going to the rich patch, they're going to the poor patch because they're expecting to find that the social group of fish was showing them the new location of the patch. So they're not taking a risk, they're going to the same location that has just been shown to them by that group of fish. They're using public information, just as we expected them to. So shy fish are using public information in both tasks, and bold fish, in fact, are completely avoiding social contact and social information entirely. one of the interesting things about studying fish shoals and groups of fish in general is that there's a lot of complicated interactions that are going on in those contexts, and it can be very hard to get your mind around, what's exactly going on, who's hanging out with who, for how long and why. Now, fortunately, we can answer some of those questions by using social networks. And by using social networks, we can formalise all sorts of indices of what's going on in that group context. Who's spending more time with who, how long, and what context. Are there particular things about those individuals, whether it be personality or sex or size or whatever, that explains some of the variation in social behaviour that we might see. So we can quantify these connections, and this is a very nice example of a social network that I just generated. And you can see here that we have individuals in this group, and the strength of the lines that are connecting these individuals tells you something about the number of interactions that these individuals have. So these thick lines, these connections here, tell you that there's a lot of interaction between these individuals, and for example, very few interactions happening between these two individuals. You can also notice that we might depict perhaps boys in blue, girls in red, or you could have big ones in blue and little ones in red, or bold ones in red and shy ones in blue, or whatever you might like. But there's lots of information that we can get by examining these social interactions, and then hopefully understand how information might flow through groups of animals. That is, does the social information flow through the individuals here? For example, the social interaction between blue individuals are quite strong. There's very few interactions between the blue ones and the red ones, so in this case we might expect information to flow rapidly through the blue subpopulation. Now, we've studied social networks in a large number of different species of fish. This is a nice example from Port Jackson sharks off the coast of New South Wales in Australia. And these are quite interesting animals. They migrate from Tasmania or all the way down in the southern half of Australia up the east coast of Australia during the winter to particular breeding locations. And they spend almost 2 months at these breeding locations in very high densities. And the same individuals come back to these same breeding locations year after year after year. They're very long-lived animals, and so you would expect that they would develop these important social interactions and social networks and preferences for particular individuals, given the consistency in which that they're spending time with them over many, many years. So to answer the question, well, do these animals have real social relationships, we resorted to social network analysis. Now, of course, following sharks in the marine environment is not an easy thing to do. We usually rely on acoustic tracking. And acoustic tracking and social networks has traditionally been done using these VR2Ws. These are long-range detectors which pick up the acoustic signal on the tags that you insert into the individuals that you're trying to track. Now, the detection range of these things is about 400 metres, which is probably too big to understand the real social interactions that are going on. So what we did with this particular paper, we looked at the 400 with the expectation that it's not going to tell us much about the social interactions, but then we used another detector called a mini SUR, which only has a range of about 10 metres. So this is the kind of interactions, this kind of scale in which social interactions are much more likely to be occurring. And we also have a proximity receiver. Now, this is actually an animal-born receiver. So it's attached to a particular individual, and it detects any tags that come within just a 4-metre radius. So this is a very individual-specific view of social interactions, whereas this one is a much more population-wide view. And in fact, these two receivers are attached to specific locations. So they tell you who is at that particular location and at what time and how often and those sorts of things. Now, the data that you get from these can be pooled into basically a social network chart which tells you who's spending how much time with each individual, and then you can generate a social network. Now, the social network that I've shown here are from a couple of different years, because obviously these sharks are coming back year after year. In fact, the tags last for 10 years. Here we have the proximity receiver in 2012. This is off one particular individual, and in this case there's really no significant assortment going on. So these sharks aren't assorting by sex, they're not assorting by size, and they're not at all interested in the genetic relatedness of other individuals. The mini SURs, on the other hand, show significant assortment by sex and by size, and you get the same result year after year after year. That's not surprising, because you tend to find these same individuals in the same locations every year. So they're pretty stable in terms of the structure of the social group. And the fact that you're getting significant assortment means that this is not a random distribution of individuals, that they're actively seeking out individuals of the same sex and the same size, but again, they're not too concerned about whether they're a cousin or a brother or whatever. Genetic relatedness really has no impact whatsoever. And the other interesting thing that these metrics can tell you is that these sexes, the males and females, tend to be equally social. So that's really important in terms of the methods that you might use in order to gather information about social networks. And there's a prediction that we might have that the information flows through these networks in predictable ways, in the same way as perhaps a disease might. In the next experiment, we actually had a look at manta rays. Now, manta rays are a very interesting group of animals, not least because they have these unique spot patterns. So rather than using acoustic technology to track individuals, you can do it visually. You just have to build up a library of individual patterns, and then you can track who you see at particular locations at particular times. And social networks and social interactions will be particularly important in a manta ray society, because they are feeding on plankton, which is very unpredictable in the spatial environment around them. And so we might think that these guys come together at fish cleaning stations, for example, where they come and the cleaner fish clean parasites off them, and they probably share information about where the best foraging patches are. And we'll just watch this movie to see how it goes.
Manta rays are charismatic species that appear to be highly intelligent and social. Social interactions are common when manta rays aggregate in large groups to feed together. Mating is also a social event, where females lead spectacular trains of males behind them. This study used photo identification of individuals to record the composition of more than 500 groups of reef manta rays. Social network analysis was used to quantify the structure of relationships within a population in Raja Ampat, West Papua. Researchers found that the rays formed social communities and actively chose to associate with preferred companions. Manta rays are threatened by many human activities, including illegal hunting and uncontrolled dive tourism. Understanding social relationships will help to predict their response to disturbance and reduce impacts on their natural behaviour and population structure.
So that tells you a little bit about the social network analyses that we conducted in manta rays in West Papua, one of the hotspots of global diversity, both for coral and coral reef fishes. Here you can see a similar sort of social network analysis that was done in wild guppies. And as I mentioned, one of the things that we want to know about these social interactions is who's hanging out with who and for how long, and whether or not this structure of the social relationships transfers into how knowledge is passed from individual to individual. And in this particular case, Darren didn't use visual ID or acoustic technology, but he invented a really ingenious net that had little pockets in it, and whenever he raised that net up, the fish fell into these little pockets, and he used those pockets to measure affiliations. Now, one of the best papers that show how information transforms or transfers through social groups is this work done by Mike Webster and colleagues, and it was in AMNAP, again using sticklebacks. And basically what these guys did is they created these groups of fishes, 8 individuals, and before they did any kind of testing or anything, they just simply quantified the social interactions using a social network approach. Then what they did is they put out a patch of prey items, of food, somewhere in an arena, and they wanted to know how quickly the identity, the location of this particular patch was transferred through the group of individuals. And they also had a control patch. This is just a random location selected to act as a control, because obviously, there's going to be fish moving around this environment all the time, and what they want to know is this a random movement over any kind of patch, or are they actually attracted to that foraging location? And they did this experiment in 2 contexts, one in a low complexity aquarium, boring open arena, and one in a structurally complex arena. And you'll see the results on this next slide. Now this is the social network diffusion information. And what you can see here is the amount of time any given individual discovers a foraging patch. In this case, the prey patch is in the top and the control patch is down the bottom. And the rate at which they discover these patches is the interesting piece of information that we're particularly interested in. So here we can see the information for the complex patch. In the boring patch, in fact, not very much interesting happened. The rate at which the individuals found the foraging patch and the random location were roughly the same, so there's not much information that we can gather from that context. But here in the complex environment, in the complex habitat, you can see that the probability of finding, or the rate at which individuals find the foraging patch, is much, much faster than it is for that random patch. Now that's really interesting, but what is more interesting is that the rate at which this information transferred through the social groups regarding the location of this foraging patch actually matched the original network analysis that they conducted on the fishes. So this is a really nice example of how information flows through a network of fishes, and it's very similar to Lucy Aplin's work that you saw in Lecture 2 on the social information of foraging patches in birds. Now, one of the interesting aspects of social learning is that it can not only occur between fish of the same generation, but it also can occur between generations. And what that means is that the information can flow from generation to generation and thus become a cultural tradition. Now, we've shown that information can pass from fish via horizontal transmission in the lab setting. For example, here the fish had to discover the location of a foraging patch, and some of them were trained to go through a green door, while others were trained to go through this red door. And what is interesting is in this technique, it's called a transmission chain technique, you have originally the entire population made up of demonstrators, but then gradually you remove those demonstrators until there are none left in that population. This is a bit like the removing the knowledgeable matriarchs, for example, from an elephant herd. So when the big female dies, all of the knowledge goes with her, and the next biggest female steps up and takes her place. And hopefully she's learnt through experience hanging out with that previous matriarch where to go, when, and all those sorts of things. So here you can see demonstrators trained to a red door. Their preference to swim through the green door is very low, so they're always going through the red door. And we removed 1 demonstrator, 2 demonstrators, 3 demonstrators, so there are now 3 naive observers and 1 demonstrator left. And at stage 5, 6 and 7, all of the original demonstrators are gone and you have 4 naive individuals, and they still have a very strong preference for the red door, and this is also true for those populations trained to go through a green door. And incidentally, you can also move the location of the foraging patch behind these doorways such that it becomes maladaptive to follow the social norms, if you like, and in which case it would be not appropriate or less appropriate to use social information, but they still manage to hold on to that seemingly random preference for green door versus red door. Now, there are lots of examples of social learning in the wild context as well, and it particularly happens where these fish are following one another to particular locations. More often than not, it's migration routes to daily feeding routes, or perhaps to breeding aggregation locations. This is a nice experiment done by Helfman and Schultz. It's fairly old, 1984, but these guys managed to manipulate entire populations of these French grunts to determine to what extent the transmission chain or cultural information was being passed down from individual to individual in the context of these daily migrations to the foraging sites and back to their resting sites. Basically what they did is they looked at the movement of resident populations. In this case, the resident population is shown in blue. During the day, they're hiding amongst these urchins, and then they make this very particular passageway out to their preferred foraging locations. And then what they did, which you can see in green, is that they transplanted some new individuals from another population into this resting group, and they looked to see what happens. And basically what happens is that those new transplanted individuals learn the new foraging location by following along the other individuals, those residents. Then they did another transplant, but this time they removed the resident population. And the results of that here are shown in red. And in fact, what these ones do when there are no residents, they go off in a random, seemingly random direction. But in fact, when they analysed those pathways, they had more similarity to that population's original pathway when they were back home than they did to the appropriate location of the foraging route in this new context. So when there are individuals there who have the knowledge, new individuals can learn about it, but when those individuals are removed, then that knowledge about the appropriate feeding migration route is lost from the population. Similar observations have been made in brown sturgeon fish, except in this case, the leaders and, more importantly, the followers do these seemingly random body movements, rolls and pitches and things along the pathway. And the followers actually imitate these movements, these dips and so on. And this is probably the only known example of potential imitation happening in a wild context. In freshwater stingrays, there was also an experiment where it looked as though some imitation was going on, and this is where the stingrays were trying to remove a food item from a piece of piping, and there was a particular behaviour that was very successful in removing that prey item from that tube, which was quite frequently displayed by the pre-trained individuals who had obviously learnt the most appropriate way of extracting the food. And the observers who had watched those demonstrators also took on that foraging behaviour, those idiosyncratic sucking and undulation movements that the demonstrators were doing. Whereas control individuals who had no experience with the demonstrators did lots of other different sorts of behaviours instead. So perhaps imitation is happening in fishes as well. And there are several practical applications of understanding social learning and culture in fisheries, particularly in the context of fisheries enhancement and fisheries conservation and management. And we'll talk to you each of these in turn. The first one pertains to hatchery production for restocking purposes. And restocking can happen either to enhance existing fisheries, to encourage new fisheries, or purely for conservation management. So you want to introduce new individuals into the population to bolster the population in the hope that they add to the breeding population of that species. The trouble is, as we talked about earlier, is if you rear fish in this very artificial environment, their behaviour is often not well suited to life in the real world. And in fact, post-release survival of hatchery fish rarely exceeds 5%. So what you can do is you can teach the fishes to learn what we call life skills, learn about foraging on live prey, learn about the identity and appropriate anti-predator responses when you come into contact with a predator. And here you can see that we were actually teaching the fish not only to recognise the prey item itself, in this case a live bloodworm, but also the likely location that this food is going to be found. And to do that, we had two demonstrators. One demonstrator was showing the observer how to feed at the surface, and the other was showing the observer how to feed on the benthos. And then, of course, you can expose the observer to either context. In this particular case, we're looking at the latency to forage on the benthos, and you can clearly see that the foraging latency for our fishes that were watching demonstrators feeding on the surface was much, much lower, was higher rather, than those that were trained to feed on the benthos. So here they've learnt the appropriate information, and here the social learning was in fact maladaptive. And of course you can do that for predator recognition type experiments as well, and indeed it has been done. The other interesting context where this is particularly relevant is in fisheries management, and particularly in wild fisheries. Now, one of the things about wild capture fisheries is that traditionally they target the biggest fish in the ocean. But this practice has unforeseen impacts if important information, like migration routes, are passed on from generation to generation via vertical social transmission. That is to say that they are culturally inherited. And in this context, the knowledgeable individuals are being removed by the fishing operations. So that leads to a collapse in the cultural information, the cultural heritage of that population, and ultimately the use of suboptimal locations, either for foraging or breeding or whatever else, and ultimately the stock collapse. And we probably reckon that this is happening in a number of different important commercial fish stocks, such as herring, capelin and cod, where they learn the locations of feeding and overwintering and breeding grounds by social learning. And because we have consistently nailed the big individuals in these populations, it has led to stock collapse. So some conclusions. Fish are much smarter than most people think. They rely both on social and private information. Social learning causes information to rapidly spread through a group, and social relationships predict how that information flows through those social groups. This is the social network analyses we were talking about. Vertical transmission of information can lead to stable culture, and understanding social learning processes has practical applications to fisheries management. Some suggested reading, and I hope you enjoyed this talk about social learning and culture in fishes. Thank you.